Nitrous oxide, along with methane and CO2, are gases which absorb long-wave radiation. The global temperature is determined by the incoming short-wave radiation and the outgoing long-wave radiation. And if the atmosphere contains gases which absorb the long-wave radiation, it works like a greenhouse. That's why we call them greenhouse gases. It stops the heat from reaching into to cosmos again.
So we are concentrating on the N2O emissions and trying to understand better why some microbial communities emit more N2O than others.
Mankind is more than doubling the input of what we call reactive nitrogen to the biosphere.
I was worried about the planet, of the state of the planet, and I thought by understanding more, that would give me a chance to actually do something for the environment.
We can trace the nitrogen coming from Europe that gets deposited in bioavailable forms onto the snowpack, and we can trace it through the seasons.
We use stable isotopes, labeled nitrogen that we can trace through systems and measure the end products that we want to.
Work is that cells in the same culture experience different intracellular concentrations of free nitric oxide, although they are in the same culture.
But it looks to me that you're running batch cultures.
Yes, maybe I specified it by saying pseudo-steady state.
So if repair is reductive, you would never know it.
No, but the point is...
How does it affect your model?
But the NO concentration as such is not modeled in the growth model. Yeah.
Denitrifying cells at the time of oxygen depletion divided by total cells, but the model was able to calculate it more precisely. F then equals denitrifying cells at the time of oxygen depletion.
There is no way we are going to reduce massively the use of fertilizers unless we want to induce global-wide starvation, because feeding the human population depends on nitrogen fertilizers.
Work with you to find out.
Spatial differences, so surface snow is completely different from the base of the snowpack in terms of chemistry.
I see, we have been expecting you.
Hello, fine to see you.
So could she not find the binding site for the reg-R?
No.
She saw a binding, but she couldn't get the actual sequence.
These applications are transferred to industry almost by stealth within months. And it's networks like this that stimulate that knowledge transfer.
These nitrogen cycle meetings have made change in the wastewater treatment industry, and I think we can make change in the agricultural industry. We need to engage with the right people now, the politicians, the agriculture managers, to make that change.
Gut people and soil people.
And mud people.
Exactly.
Yes.
There are many aspects in research, and some is that you have some lonely work to do and struggle with writing.
I focus on the characterization of one enzyme, and here I can have contact with the researchers that work at a microbiology level.
When you have multidiscipline and multi-styles of researching, then you can help to put your own research into a better context.
Europe is the only place where a nitrogen cycle has been properly studied. I have no doubt.
We have so many new tools to use nowadays with the molecular methods, with isotope work.
Probably no one in this business of global warming research can claim that they alone solve the problem, but we make small contributions.
We have a fantastic network in Europe who have been working on these questions for quite some time.
We can probably make the food production cleaner in the sense that we can manipulate the soil in a more environmental friendly way to reduce the fraction of the nitrogen which goes back into the atmosphere as N2O. And this is our target.
Follow your heart.